All right, so what we're going to do today is we're going to go through some of the highlights from lecture 7 and 8. So this is not meant to replace going through the detailed lectures, but to hit some of the highlights like we normally do in class time with it. So I will be skipping over some things in here. So what we're going to look at first is the cell membrane, the structure and function of it. So they're amphibolic, meaning they've got both hydrophilic and hydrophobic regions, and these are key to their structure. So you're going to have hydrophilic regions that are on the inside surface and the outside surface, and then you're going to have hydrophobic regions in the middle. And this is going to allow it to interact with watery substances on both the inside and the outside, but the watery substances don't like to cross through that middle area. So it's a fluid mosaic. So you're going to have these things slide around past each other. You're going to have proteins embedded in here that are going to have these other pieces sliding around and moving them. So the membrane is fluid. It is dependent on temperature. So one of the reasons that we do have to live at certain temperatures is if it gets too cold, the membrane is going to start to solidify too much. If it's too warm, the membrane will liquefy too much. So cholesterol acts as a little bit of a buffer in there. So it restrains movement so it doesn't get too liquid, and it keeps it from packing too tightly so that it doesn't get too cold, so that we don't have to live at quite such a narrow temperature range, but there is still a definite temperature range in there. So with membranes, they are going to have a lot of proteins in there. There's over 50 proteins that have been identified so far on red blood cells alone. So when we look at the categories of proteins, you can have integral proteins that are going to penetrate into the hydrophobic core. Transmembrane proteins go all the way across. Peripheral proteins are going to be appendages on the outside. And then the integrins are going to help attach it to the extracellular matrix. So we're going to be looking at different functions of the membranes. So its structure does allow for selective permeability. So the nonpolar hydrocarbons, carbon dioxide and oxygen, are going to be able to cross easily. The hydrophobic core is going to impede the passage of ions and polar molecules. They're not going to want to cross quite so easily. So in looking at the types of proteins we have, one type is transport. And these are going to allow specific ions or molecules to go across. So with a channel protein, it's going to have a hydrophilic channel that certain molecules or ions can go in. Aquaporins are going to be ones that are specific to water. And then some are going to be carrier proteins. So they're going to hold on to that chemical passenger, and they're going to change shape when they go across. So those are specific for translocation. So when we look, they can see that it is transporting something across. Here, you can have them act as enzymes. They can be part of signal transduction to pass communication on in the cells. They can attach to the extracellular matrix. They can join cells together or be used for recognizing things. So when things move across, a couple things we're looking at is energy involved with this? If energy is not, it's a passive process. So when we look at diffusion, it's going to be molecules moving across. Here, their goal is just to spread out. So we've got this membrane here. You've got these molecules of dye, and they're able to get across the membrane. So what they're going to do is they're going to spread out till they're equally spread out on both sides of the membrane. So they're going to move down the concentration gradient. They're going to go from the area that's most concentrated to the area that's less concentrated. If you have two different dyes, they're going to go in the different directions they need to so that they will each independently be diluted on both sides. So they want to diffuse and spread out equally on both sides. When we look at water, water is going to be the osmotic balance. So that's looking specifically at water moving. So in this example here, we've got these molecules that are too big to get across this little membrane. So if you can't move the sugar across this membrane, what's going to happen is some of this water here is going to move across the membrane to dilute the other side so that things are still equally spread out. So tonicity is just the ability of a solution to cause a cell to gain or lose water. Ones that are isotonic, it's the same on both sides, so there's no net movement. If it's hypertonic, the cell's going to lose water to the environment. So we say that solution is hypertonic, then the cell is going to lose water to try and dilute the solution. The cell ends up shriveling up and dying. That's what you would have is if you put a human cell into really concentrated salt water, it's going to pull some of the water out of that cell and cause it to die. If a cell's in a hypotonic solution, the water is going to go into the cell and try and dilute what's inside the cell to match what's in that solution, and it will swell and burst. So we can see here with some examples of an animal cell, when it's in a hypotonic solution, it swells and bursts. An isotonic solution, it's going to stay the same, going to be a normal shaped red blood cell. And then if you put it in a hypertonic solution, it's going to shrivel up. Plants are a little bit different. They like to have this turgor where they're going to have pressure that pushes back out against the cell wall. So if it's in a isotonic solution, the plant's going to be flaccid. But turgid is actually going to be a good thing for a plant. It's going to be in a hypotonic solution, so that water is going to come in and keep putting pressure against the cell wall. So that's going to keep that plant nice and firm and strong. If it's in a hypertonic solution, it can become plasmolyzed, where you start to have the plasma membrane pull apart from the cell wall, and it's going to wilt. So facilitated diffusion is going to be aided by a transport protein. So it's going to have a specific location that it's going to cross to move things over. So here you can see you've got this channel protein that's going to move things. So sometimes they can have different gates on them that will determine when they open and close. So you can have sodium channels that are going to have ion gates that will respond to the level of ions. So for active transport, these are going to require energy. The sodium potassium pump is a huge one of these. So what's going to happen is the sodium is going to come in and it's going to bind here in this little transport protein to the little receptors in there. It's going to require energy in the form of ATP. So what that will do is cause this to change shape, and it's going to release the sodiums to the outside. Then what will happen is potassium will come in. When it releases that phosphate, it will change shape and move the potassium inside. So that actually is a significant amount of energy that your cells spend on a regular basis. So these are important because they can help cells maintain a membrane potential. They'll have voltage on them like a battery does. So the membrane potential is just the voltage across the membrane where those charges are separated. So here you can see where you've got this proton pump. This is an electrogenic pump that is going to pump hydrogen ions out. And those will be used for ATP synthesis when they want to cross back over the membrane. So co-transport. With co-transport, you're going to have ATP that is going to pump something directly across that's going to want to come back. And so when this comes back, it's going to take sucrose with it. So it's going to co-transport the hydrogen ion and the sucrose together. Sometimes things are going to be too big to go across the membrane. So that's when we use exocytosis and endocytosis. So exocytosis, think of the X as things exiting the cell. This is going to allow bigger items to come out. So with a lot of the secretory cells, they'll do this to release insulin. Your neurotransmitters are released this way. Endocytosis is going to take in the bigger stuff. So phagocytosis is the term we use for cell eating. It's going to engulf the large particles. It wraps the pseudopodia around it. So you can see here in this lower picture on the left that it's engulfing that particle around it here, and then it will take it into a vesicle. Pinocytosis is when it's going to be droplets that will be taken in. And then receptor-mediated endocytosis, this allows bulk materials to come in and for them to be concentrated. So with these, it gets one little particle in or one little droplet in and it swallows it. With this, it's going to wait till all of these receptors are full, and then it's going to swallow the concentrated vesicle of things at that point. So we're going to move on to looking at an overview of metabolism. So next week, we'll be getting into more of the details with metabolism. This is just kind of the thermodynamics of it. So metabolism is all of the chemical reactions that go on in a cell. However, we usually are focusing on the ones that are used to derive energy. We study them in pathways because otherwise, if you learn the individual reactions and they're in random order, they're going to be really unpleasant to work with. So that's why we do it that way. So the catabolic pathways, these are going to be ones that break things down and release energy, where the anabolic pathways are going to be ones that are going to have to consume energy. So we've talked about energy before. It's the capacity to cause change. Kinetic energy is the energy of movement. Heat falls into this category because you're going to have atoms moving around. Potential energy, this is due to location and structure. Chemical energy is an example of this, where you've got atoms that are bonded together and there's potential energy to be released from the chemical reactions they do. So you can see here, this person has potential energy because of their location. This one is exhibiting kinetic energy with their actual motions. Thermodynamics is just studying the transformation of energy. We look at things in a system. Everything outside the system is the surroundings. So isolated systems do not exchange with their surroundings. They're actually pretty rare. An open system is going to be where energy and matter can transfer between the system and the surroundings. So it's very difficult to create a closed system that's isolated. Almost everything is open because we can't stop things from reacting with the air and the environment. So the first law of thermodynamics says energy cannot be created nor destroyed. It's transferred or transformed. This is that conservation of energy. The second law is going to say during that transformation, some of it's going to become unusable. So a lot of times it's going to end up being heat, and it makes the universe more disordered. So when you think about it, when you get gas and you're running gas in your car, some of it's going to be used to move your car, but some of it is going to just be wasted as heat. So if it's really cold outside, you can see that the frost will melt on your car because it's generating heat. That is some of it becoming more disordered. So entropy is this measure of disorder or randomness. So anytime you have a spontaneous reaction occur, it's going to automatically increase the entropy of the universe. So when we look at free energy, it's the amount of energy that can be used to perform work. There's different components to it that make it up. So in chemistry, you do more of the calculations of these things. In here, it's just that you want to know the components of free energy. It's going to be made up of the change in enthalpy, the change of entropy, and the absolute temperature in Kelvin. So we call this free energy a delta G. The little triangle symbol here is for delta. It has to be negative for it to be a spontaneous process. So the reason for that is delta G is actually made up of the free energy in two different states. So when you take the energy from the final state and you subtract the energy from the initial state, that's going to give you what the delta G is. So delta G is negative when there is a loss of free energy. It's going to be moving towards a more stable state with a lower delta G. So as things move towards equilibrium, the free energy is going to decrease. And as it moves away from equilibrium, it will increase. So there's a couple of pictures that will help illustrate how you come up with this delta G that we'll look at in just a minute. So in an exergonic reaction, this is going to be one that is going to release energy. So I just remember it with the X is it's exiting. It's going to have a negative delta G. So if we look at the reaction for cellular respiration, where you've got glucose reacting with oxygen to make carbon dioxide and water, it's going to release 686 kilocalories per mole. So that mole is 180 grams. That's under standard conditions. So your products are going to have 686 kilocalories per mole less than the reactants did. An endergonic reaction is one that you're going to have to put energy into. So the delta G is going to be positive. So here, these pictures are helpful in understanding the difference where you come up with this delta G. So if you've got an exergonic reaction here, this is going to be one where energy is released. It's spontaneous. Your reactants are way up here with their free energy. The reaction takes place and then the products are way down here. This difference in them is going to be the amount of energy released. So the products here are down here. So when you subtract your reactants from your products, that's how you get the negative delta G. So with an endergonic reaction, this is not spontaneous. You've got the energy of the reactants down here and the reactants of the energy of the products way over here. You've got to put energy in to get these products up here. It doesn't just happen by itself. So this is the energy that has to be put in. It'd be a positive delta G. So with metabolism, these things are always working towards equilibrium, but you really just don't ever achieve equilibrium in a living system. There's always going to be something that is being consumed, pulling the reactions in one direction. So with these reactions, we're going to be creating ATP, and ATP is used to do various types of work. But we're going to do it in a small amount of steps. So if you look here, this is showing an isolated system. This allows the water to actually reach equilibrium on both sides. That's not going to happen in a living system. With a living system, you have a constant draw here. So it's always going to need to have something come in, and it's never going to quite reach equilibrium. And with our metabolism, it's actually multiple steps like this that we're going to go through so that you're constantly moving things down. That's why you have so many different reactions in metabolism. So next week when we're going over that, you're going to be looking at all the steps going, why so many steps in here? And we do this so that the energy is released in small amounts and it's usable for the cells. So how the cells are going to release energy is they're going to store it in ATP. So it's adenosine triphosphate. It's got a ribose, adenine, and 3 phosphates on there. It works well to store energy because what's released is usually greater than most other molecules involved. So in a lab, it's going to have negative 7.3 kilocalories per mole under standard conditions would be its delta G. But the cell is actually perfectly set up for this. So under cellular conditions, it's actually going to release 78% more energy. It's going to actually be 13 kilocalories per mole. So this is specifically set up to work for our cells. So this is just a model here of what ATP looks like. I actually prefer this bottom one in here. I think it's just simpler. So you've got your adenosine triphosphate. There's the 3 phosphates on it. You remove one of them, it will now become adenosine diphosphate. So it's ATP, ADP, and it's the difference of a phosphate being taken on and off the molecule. So here you can see it working with this transport protein here. It's got this one orientation here when it's got a phosphate on it. When you pull the phosphate off, it's going to change shape and it will switch its orientation to help transport things. It can work for transport vesicles. So these will actually have the ATP going on and off of them. And as they do, it causes them to kind of move along the cytoskeleton tract. So we will be looking at those reactions that you use to regenerate that ATP. So we have all these reactions going on. We need to have a way of controlling them. And that's where enzymes come in. They're going to act as a catalyst that will speed up the reaction, but enzymes are not confused or consumed. So when we look at them, you're going to have different energy levels here. You've got the energy of your reactants, the energy of your products here. The difference between these two is delta G. Enzymes don't actually affect the delta G at all. But in order for the reaction to take place, a little bit of energy has to be put in here. And we call that the activation energy, that it will get it over this transition state. So that's what keeps these from just going spontaneously by themselves all the time. So what an enzyme is going to do is it's going to lower that activation energy. So it's a smaller hump that it has to go over. However, the delta G is not affected. So the difference between the reactants and the products is still the same. It's just not as much of a hump to get over it when you have enzymes involved. So enzyme naming is actually really logical. It ends in an ASE ending. So if you've got sucrose and you want to break it down, the enzyme's going to be sucrase. If you've got a peptide that you're going to break down, it's going to be called a peptidase. So with enzymes, the substrate is going to be the reactant that the enzyme is going to act on. And when it comes together, it's going to make this enzyme substrate complex. Where it has to go is this active site, and that is specific for it to fit in there. It has an induced fit, so it's almost like it kind of hugs it a little bit, and that puts it in the right position to be able to be doing the catalysis. It's going to be very brief that it holds that. So a single enzyme can act on thousands of them per second, so it's very quick going in and out. So when we look at this, the enzymes, their rate of function is partly going to be related to the initial concentration of a substrate. So if you think of it as a substrate's like a toll booth and you've got 3 stations, if you bring in 1 car per minute, you're going to get 1 car per minute out the other side. If you bring in 2 cars per minute, you're going to get 2 out the other side. If you bring in 3, you'll get 3 out the other side. If you bring in 6, you're still only going to get 3 per minute out the other side. At some point, things become maximized and they can't go any faster. And it's going to be the same thing with enzymes. They can only function so fast. So you can only make things faster by saturating them up to a point, and then they can't take any more and go any faster. Enzymes have optimal conditions they work in. So they like to have the correct temperature. When we look here, some enzymes like to be at 37 degrees. For some of the thermophilic bacteria that like to have warmer temperatures, their optimal temperature is going to be 77 degrees. So they really don't have an interest in living in our bodies because our body is not going to be warm enough for their enzymes to work. Enzymes also tend to have an optimal pH. So things that in our stomach, like pepsin, it has to function at about a pH 2, but there's other enzymes later on in the tract, like trypsin, they need to have things be about a pH of 8 to work. That's why you can't just mix them all together in one place. If you did that, you wouldn't have the right pH for either of the enzymes to work. So cofactors, these are going to be non-protein helpers. So a cofactor would be something inorganic like a metal or a mineral. That's one of the reasons why they're so important in our diet is they act as cofactors in there. Coenzymes are the organic ones. So these would be your vitamins. When we look at metabolism, B vitamins are actually a very common cofactor for a lot of those enzymes. Then we have enzyme inhibitors. A competitive inhibitor is going to mimic the substrate. So it's going to compete for that same place that the substrate uses. So if you put in more inhibitor, you're going to slow things down. If you put in more substrate, you're going to overcome that. With non-competitive inhibitors, though, they're going to bind to another place. And it doesn't matter if you add more substrate to it, that's not going to overcome that because it's bound and locked up in another place. So a lot of toxins and poisons are actually irreversible enzyme inhibitors. So sarin nerve gas, DDT, some antibiotics actually function that way. So some of the enzymes can actually work together. So they will have activation on an allosteric site. So sometimes you can bind one active site and it will actually stimulate catalysis of a multi-unit enzyme that will affect other active sites. Sometimes they will be cooperative in there. So if you cause an induced fit in one subunit, it will do the same thing in all the other ones. Hemoglobin does this in the vertebrates. So what will happen is once it binds to an oxygen in there, it's going to change the shape a little bit and it's going to make it more favorable. So it's going to want to bind oxygen even more. And that will help you get more of the oxygen onto the substrate, onto the enzyme rather than having it just floating around in the bloodstream. So feedback inhibition is one of the ways that we can shut down enzymes. So here with this example, you've got your substrate that's going to bind to this enzyme here and it makes this product. What the product will do is if it starts to build up, it will actually inhibit the enzyme and shut it down and say, stop it, we don't need to make any more. So it's a good way for the body to be able to regulate and only make what it needs. So that is your quick overview on this. So you are certainly welcome to email me with questions on things this week, but I just wanted to get this out to you so you had something to study with and give you a good overview going through things. Thanks.